All right. Okay, good. So it is a great pleasure to introduce Marie-Constance Corsi. Let me go through a quick bio of Marie-Constance. She's an INRIA research scientist at the Paris Brain Institute in the NERV Lab. Her research focuses on the development of tools to address the BCI inefficiency or illiteracy, as we all know it, problem, reflected by a non-negligible portion of users who cannot control BCI devices even after training. She essentially considers two main approaches to tackle the problem: the search for neurophysiological markers of BCI training and the integration of multimodal data to enrich the information provided to the classifier. In 2025, she received the very honorable Early Career Award from the International BCI Society. She has previously served as Secretary General of the French Academic Association promoting the advances of BCI, called CORTICO, and she has been also the co-chair of the Postdocs and Students Committee of the BCI Society. And with this, I will just pass the floor to you. And the title of today's webinar is Investigating Brain Interactions, a Dual Path to Understanding and Improving BCIs. So, MC, whenever you're ready, you can start.
Thank you very much. Good afternoon, everyone. Thank you, Simis, for these kind words and for having me today. It's a pleasure to talk with you a bit about my research. Perhaps before starting presenting what I'm doing, just a bit of elements of context about my background, because I don't have a super linear journey. So just to start with, after the engineering degree in telecommunication, I embarked on a PhD focused on the development of alternative MEG sensors for magnetoencephalography sensors called optically pumped magnetometers at CERN in Grenoble. And at the time, the objective was to eliminate the cryogenic cooling system that you can see here on the screen, that are currently developed in the current MEG system. And the aim was to develop miniaturized and more affordable system that could reduce the distance to the sources of activity. And so we experimentally validated the prototypes that could record the magnetic field either generated by the heart here at the beginning, and then we also validate them this time by trying to record the brain activity. And so, yes, this is my second home here, as you can see, because I've made many experiments in this lab. And yeah, at the end of my PhD, I wanted to find a domain that could combine my engineering background and the neuroscience, and therefore it was quite natural for me to apply for postdoc position on brain-computer interface at the ACM, proposed by Fabrizio Di Falani in collaboration with Daniel Bassett from Penn University. And this postdoc at the time focused on BCI training in healthy subjects and stroke patients, involving a significant amount of experimentation, which I love, and also analysis. And also it enabled me to interact with clinicians, technicians, and more importantly, patients and their families. And so during this project, I noticed that my colleagues perceive me differently depending on their backgrounds. For instance, it was especially evident when I have to deal with clinicians during the conception of experiments, but also with machine learning and deep learning aficionados when I try to convince them to investigate the interpretability and explainability of their models from a neurophysiological perspective. So my research here reflects these aspects, and indeed, to address BCI limitations, I am particularly interested in the interactions between the machine and the subjects. So yeah, the title of this slide, I borrowed the expression from the paper by Jose and Simis, as in my opinion, it accurately reflects the mechanisms occurring while the user tries to control the BCI system. And we all, I think, have observed notable inter and intra-subject variability to control such devices, even after several training sessions. So historically, people have adopted a machine-centered approach to improve either the quality of the signal or the classification tools to decode the subject intent. But more recently, there has been also a growing interest in developing user-centered approaches that consider human factors, cognitive profiles, or also potential neurophysiological markers of BCI performance. So in both cases, they're perfectly relevant. but they mostly rely on local measurements, disregarding the exchange of information between areas and therefore the interconnected nature of brain functioning. So here I'm interested in investigating to what extent brain interactions can inform and potentially improve the use of BCI. So yeah, again, before presenting my research, some elements of context and definition, perhaps. So here, when I'm talking about functional connectivity, the idea is to start by working from the signals that can be captured by different modalities invasively or non-invasively way. We can take the example of EEG, for instance. And once you have captured the signals, you can extract what we call a connectivity matrix, for which each element will correspond to a statistical assessment of the interaction between two elements. That could be two electrodes, two brain regions, depending whether you're working at the sensor or at the source level. And to obtain such matrix, you have plethora of functional connectivity estimators depending on properties such as whether you're working at the frequency domain or in the time domain, whether you're interested in directed or non-directed estimators, or whether you prefer model-free or model-based estimators. But in the end, you obtain such matrix. And based on that, you can binarize it to obtain threshold it to have like one single network or a graph, for which you have the nodes representing the brain areas and the edge corresponding to the potential interaction between two nodes. And once you have this network, you can investigate the different topological properties of such network. And perhaps the most intuitive property would be the degree that corresponds to the sum of the edges associated to a given node. And such properties can be used for different purposes. You can use such information either as a potential feature for the classification, but you could also use this information this time as a potential biomarker to investigate mechanisms underlying the BCI performance or even BCI training. So during this presentation, I'm going to illustrate both of these approaches. I'm going to start by the machine-centered approach, so by the classification itself. So here, the idea is to potentially consider functional connectivity as a feature. My point is not like to replace what has been done so far, but more about enriching the information of interest. And for that, it is quite linked to a not so new idea of hybrid and multimodal BCI, for which in the case of hybrid BCI, you're going to combine, for instance, EEG with biosignals such as muscle activity, and with multimodal BCI. Thank you. And for the multimodal BCI, you have to combine different neuroimaging approaches. And so here, my first attempt to combine these different modalities was through data I collected in my postdoc, for which I had both EEG and MEG data. And for that, I used one classifier per modality, and that I directly combined through a linear combination that relied on weights attributed to each of this modality that relied on the posterior probability. That is quite linked to the idea of hybrid BCI also proposed by Lib a few years back. It's quite similar architecture, but this time by combining EEG and MEG. Perhaps you can notice that we have two different submodalities for MEG. It is linked to the electrodes you're working with, because in MEG you have magnetometers and graduators. In the case of magnetometers, you're going to measure the magnetic field generated by the brain, and with the gradiometers, it corresponds to the gradient of the magnetic field. And since it's because I'm coming from physics, so I was quite shocked in mixing all this information. I preferred having a specific classifier for each of these types of electrodes. And once we have this prediction, so in this case, we worked with motor imagery-based BCI. So we had to predict whether the subject was performing a motor imagery or whether they were at rest. We could estimate the area under the curve, and we could compare the performance across different characteristics. First, the number of features. And in this case, we did not observe an effect of this number of features, but we observe a significant improvement of the AUC with the fusion approach with respect to the other modalities, in particular the EEG, that is a golden standard. Such difference in terms of performance was a bit less different in the case of gradiometers, because since it's a gradient, yeah, we can speculate that you have a higher signal-to-noise ratio. So since it is a bit less noisy, probably you have a higher performance due to that. So it was my first attempt in trying to combine different types of information. But based on that, I was wondering whether we could generalize this approach this time by trying to using heterogeneous features. And that brings me to a collaboration I initiated with Sylvain Chevalier from Paris-Saclay University and Florian Heger from INSA Rouen. So the idea was that currently in the BCI, you have these geometric methods that rely on Riemannian geometry. They use covariance matrices that can capture some invariance from the EEG signal. They provide good generalization, they're compatible with online requirements, and more interestingly, perhaps also, they are robust towards noise and to small changes in the placement of the EEG electrode that could happen during an experiment. And on the other side, we have also the functional connectivity. And in the team, we have previous works led by Tsnakatai and Igor Carrara and Bruno Ristimunia. They all proved that functional connectivity could be used as potential features for the classification. So based on both of these aspects, our hypothesis was that combining functional connectivity estimators and Riemannian geometry will lead to an improvement of the classification performance. And so we first tried that during a BCI competition a few years back. And the idea was to classify data obtained from stroke participants who had to perform motor imagery tasks. We ranked first for one of the categories. We're quite happy of that. But it was not the end of the story, because we did not want to provide like a pipeline that could be like one-shot thing that could work only on this data set. So we tried to optimize our approach and to thoroughly also perform a benchmark with the state-of-the-art algorithms. And with that, we arrived with our approach called Vougon. It's a Corsican word. If you want, I can explain afterwards what it means. But in short, the idea is to combine both of these approaches. So we have the EEG signals, and we are going to apply two first-level classifications. So in the first branch, we're going to extract from the EEG the covariance matrices that are going to be projected onto the Riemann manifold. And then we're going to apply a prediction. Here in our case, it was based on elastic net. On the other branch, we're going to estimate the functional connectivity that are also going to be projected onto the manifold. We also ensure that the matrices were SPD, and we also applied some regularization if needed. And then we could also perform a classification. And in the end, we applied an ensemble learning approach to be able to combine both of these classification outputs in a single prediction. So again, since our aim was not to do one-shot thing, we perform a quite thorough, I would say, benchmark on a large number of data sets and different configurations. So we assess the performance of our approach from a 2-class problem to a 4-class problems in within and cross sessions. So I'm going to just illustrate my point with one given example taken from a 2-class problem that was, if I'm not correct, right hand versus feet motor imagery. So don't be afraid by all these pictures. So we can start locally just by the first one. So the idea was to, in each case, to compare our approach, Vougon, with another pipeline taken from the state-of-the-art. So we compared our approach with methods based on spatial filtering associated either with LDA or SVM, but also we compared an approach with Riemannian geometry approaches, the classical ones. So let's take the example of the first picture at up left. So here the idea was to assess our performance across different data sets. Here we had 5 data sets. And for each of them, we assessed the performance and we statistically compared our performance with another pipeline. So we have here the Vougon approach, and we compare it with the CSP, the regularized CSP associated with shrinkage LDA. And we did this statistical comparison for each of these data sets. And in the end, we could have access to a meta effect to see whether our approach could robustly improve and be outperform perhaps the other ones. Such kind of analysis has been performed for all the different one versus one comparison. And it was made possible thanks to the MobiB, that is a Python toolbox that I strongly recommend. I'm not paid by them to say that. Actually, it gives you access to a large number of publicly available data sets in BCI, not only on multi-imagery, because it's true that it's my main research, but also SSVP, any types of BCI paradigm. And also it provides you some guidelines on how to benchmark your approach with the state of the art. So I think it's a good for your research, but also as students, I think it's a good way to learn how to perform this kind of analysis. So our approach has been made available through different papers, also GitHub repo, and we also made a tutorial that is available through the Powerman Python package. So feel free to look at it and to try it out on your own dataset if you're interested in. So this kind of approach could enable us to demonstrate that it could outperform the other pipelines. But then, since we previously saw that we could combine EEG and MEG, what about generalize it a bit more and also combine this approach this time with MEG and EEG? And it corresponds to this work in which I did the same kind of pipeline, but this time adding another two first level classifiers based this time on MEG. And we could also demonstrate that having this kind of ensemble learning approach could improve the performance. Here I worked in the source space, for instance. So here in this first part of the presentation, I showed you some illustration of the use of functional connectivity as a potential tool for the classification. Now, in the second part of the presentation, I'm moving a bit more towards the neuroscience and to see whether functional connectivity can be used this time to investigate potential neural mechanisms underlying the BCI performance and training. And for that, I'm going to rely my presentation on the dataset I collected during my postdoc, for which we conducted a BCI protocol that consisted of including 20 HACI participants, all BCI naive, is an important point. They had to come 4 times, twice in a week during 2 weeks. And during the experiment, they had to perform a multi-imagery task. So here is a video on the right, corresponding to one of our BCI experiments with one of our included subjects. During the experiment, we simultaneously recorded MEG and EEG data, even though during the online classification, it relied only on the EEG data, just to be perfectly clear. In this case, the instructions were dependent on the position of the target displayed on the right part of the screen. And the aim was to hit the target with the moving cursor, corresponding to the feedback in our case. So here, only the vertical position of the moving cursor was controlled and was modulated through the cerebral activity. So when the target was up, the subject had to imagine a movement with the right hand. And when the target was down, the subject had to remain at rest. And in my case, since I collected data, I performed the preprocessing and I worked directly in the source space. So my first work consists in trying to see whether functional connectivity could be relevant to identify potential areas of interest and whether they could correlate with the BCI performance. It's simply that before trying to identify some fancy biomarkers, it's also important to ensure that we actually observe a BCI training effect, because sometimes there's not a clear guideline of how many times you need to perform such training sessions, neither the frequency between 2 sessions. So it's important to do this kind of sanity check. So for that, I relied my analysis on the BCI score that corresponds to the percentage of trials in which a subject could hit the target with the moving cursor. This is not a flawless criterion, I need to say that, but is a way to assess whether we could observe a training effect. So here, we can observe the distribution of the BCI score across the participants for each session. And again, as you can see, we have a strong inter-subject variability. It's not a big surprise, but it's also important to mention that. And when we perform a one-way ANOVA, we could observe a significant improvement of the performance across the sessions. So that can help us in saying that we actually observe a triggering effect across the sessions. The second sanity check we performed consisted of investigating changes of the brain activity across the sessions. So for that, again, working in the source space, in this case, I worked with EEG here at the group level. And we compared the power spectra obtained in multi-imagery versus rest, and we performed t-tests at the group level. So within the high alpha beta band, high alpha band, sorry. So here at session 1, we didn't observe any significant effect, which is not again a big surprise because these participants were completely BCI naive. It was like the very first experience of BCI. But as time goes by, we can see first a quite diffuse desynchronization effect occurring here, and then that tends to be a bit more focused over the left sensory motor area. Again, it's not a big surprise because it's quite in line with the literature, but it was important for us to make sure that our observation and the neurophysiological signatures make sense with previous literature. Another question also we wanted to ask was whether the improvement of the performance was due to a learning, but which kind of learning? Do the participants learn how to remain at rest or to perform the motor imagery task? So for that, we compared the power spectra obtained within the beta 1 band, so the low beta band, either at rest or at motor imagery. And either we did not observe a significant time effect in resting state. Such effect was a bit more important in motor imagery, meaning that this improvement of the BCI performance seems to be driven by the motor imagery task itself and the learning how to perform the motor imagery task. So once we have performed all this sanity check, we can now investigate properly and we can work with functional connectivity. So for that, we worked with knot degree. So again, it is a network property that corresponds to the sum of the links associated to a given node. So in this way, we have one value per region of interest. And we could observe two things. First, the involvement of sensory motor areas. Again, not a big surprise given the task performed by the participants, but also the involvement of associative areas and in particular, prefrontal areas known to be involved during decision-making process. It is not completely a surprise because since they were all BCI naive, they had to pick the right strategy to perform the motor imagery. That is why we may expect some process to pick the right strategy in a robust way along the sessions. And interestingly, we observe this significant correlation here in red with the BCI performance of the same session, but also we could observe a significant correlation between the knot degree at session I with the improvement of the performance in the subsequent session, which was corresponding in our case to a learning rate. And so this kind of observation actually has been made in EEG, in MEG, and also by combining MEG and AEG through a multiplex network. So it was also a way for us to replicate our observation and to make sure that we always observe the same effects. And we speculate about a potential reserve effect, meaning that where we have a higher connectivity, we expect also to observe a higher potential to disconnect while learning. But actually, I chose also to hide one teeny tiny thing. That is, so far, I am working, for instance, with imaginary coherence or coherence-based function connectivity estimators. And these methods rely mostly on the assumption of linear interactions between brain signals, either for simplicity or also to provide a more intuitive interpretation. However, they may fail to provide a complete description of the temporal properties of signal interactions. Therefore, I wanted to go one step further and to see whether other estimators could enrich the observations made so far, and in particular, by trying to capture non-linear information. And for that, actually, I initiated a collaboration with Pier Paolo Sorrentino, who is a neurologist who has investigated a lot non-linear brain interactions through the lens of the neural avalanches. These are periodic and spontaneous bursts of enhanced activity observed across various imaging modalities, not only EEG and MEG, but also across different spatiotemporal scales. So here is a video obtained from MEG data at resting state in the case. And our approach is grounded in statistical mechanics, suggesting that long-range correlations emerge in near-critical dynamical regime. And criticality in complex systems is characterized by fluctuations without any privileged spatial or temporal scale. And yes, previous works also in the field of criticality showed interesting markers, especially in resting states and evoked responses. So here our hypothesis was that neural avalanches could spread differently depending on the task, and that could also provide potential new markers of BCI performance. So for that, I'm going just to spend a few minutes just to explain you how we can obtain this ATM that refers to avalanche tracer matrix. And then I'm going to illustrate my point from data obtained in BCI experiment. So here the idea was, since we wanted to test for potential differences between motor imagery and rest, we wanted to see whether such differences could be observed from the probabilities of difference in terms of perturbations. And so we use the framework proposed by Per Paulo a few years back. So based on the signals we can capture, for instance, in EEG or MEG, with these scores and threshold, in our case, each source reconstructed signal. And from that, we could elicit some neural avalanches. So here we have the signals. We can z-scores data and apply the threshold, and we obtain a raster plot. So we have like kind of binarized information where you have 1 or 0, whether you detect an activation or not. And so we identify an avalanches that starts when at least one brain region shows an activation, and it will last when the last region is activated. And based on this raster plot, you can obtain the ATM, for which the IJF elements corresponds to probability of region J to be activated at time t+1, knowing that region I was activated at time t. And so when I'm talking about ATM, actually meaning this kind of matrix are going to work with in the subsequent slides. So before trying to see whether it could be relevant for BCI, the first question would be whether we could distinguish the task performed by participants. And for that, for each subject, we statistically compared the ATM obtained in motor imagery and rest. And such analysis has been performed for all the participants. And we observed that these avalanches involved these two premotor areas in the huge majority of participants in a quite reliable way. Whereas when we perform such analysis with power spectra, we failed in obtaining such reliable patterns across participants. And so based on these observations, we're wondering whether we could use the ATMs as potential alternative features for the classification. So for that, we build a pipeline consisting of using the ATM, and we use a support vector machine as a tool for classification. Again, our aim was not to have the fanciest classification tool, but just to see whether the ATM could be a relevant feature for the classification and could discriminate or not the task performed by the participants. And we compared our pipeline with a standard one that relied on spatial filtering associated with support vector machine. At the group level, we observed a reduced inter-subject variability, but perhaps more interestingly, in my opinion, would be the last figure on the right part of the screen, for which here each dot corresponds to the performances obtained for each participant. The X coordinate corresponds to the average performance obtained by our approach across splits of the cross-validation, and the Y coordinate corresponds to the average performance obtained with the standard pipeline. And the color codes for statistical significance when we compared performances obtained from a single subject between our approach and standard one. And we can observe a huge majority of Salmon dots, meaning that in the huge majority of participants, we observe an significant improvement of the performance with our approach as compared to the classical one. So that was a way to see to what extent the avalanches could be interesting in our case for classification, but it's just part of the story because also we can use the avalanches this time to investigate processes during BCI training. And it corresponds to the work led by Camilla Manino, who is a brilliant PhD student I have the pleasure to co-supervise. She investigated the physical properties of the avalanches across time. In particular, she was interested in the average avalanche length. She observed that at the beginning, she studied the distribution of such information across participants. So in blue, have the distribution in the rest condition, and in red, in the multi-imagery condition. At the beginning, again, since the participants are BCI naive, we don't observe a clear difference between the two distributions. But as time goes by, we have in the end a very clear distinction between the two distributions. Interestingly, she could use such information and she could observe a clear correlation with the BCI performance. And the ultimate point was that she was able to use such physical properties from the first 3 sessions, and she was able to predict the BCI performance of the last session based on that with an accuracy of 0.91. This is quite good. So in the end, it means that the avalanches could be quite interesting to predict the performance of the subsequent session, and we hope so in the long run, perhaps adapt a bit the training program based on this first baseline session, if I may say, to provide perhaps a more user-centered training program. As perhaps on the side, I also have to mention another way to use avalanches, this time for diagnosis. It is not really related to BCI, but why not? We can still mention that. For instance, we could use avalanche this time to distinguish groups. With Gianmarco Duma, we were able, for instance, to distinguish healthy participants from epileptic patients. And more recently, with Pierpaolo Sorrentino, Dimitri Paszewski, and Maro Roca, we're also working on differential diagnosis to try to see whether such information obtained from functional connectivity could help us in discriminating different neurological disorders. So it's still a work in progress, but we're still interested in this kind of topics. So in this presentation, I showed you different ways to use brain connectivity, either as potential features for the classification, but also as a potential biomarker to understand behavioral or simply cognitive processes occurring while doing controlling the BCI system. But I think that a skeptical mind could tell me that so far I presented offline work. So what about trying to translate it for BCI research? So 2 elements I would like to mention. The first one would be that we are trying to translate it from different perspectives. Perhaps the first one would be from the hardware perspective. In this case, I would like to mention the great work led by Tristan Venot. He did his PhD with us, and he developed a BCI platform that relied on a robotic arm because he was interested about the timing to provide the feedback to the participants with this robotic arm. And such findings enabled us now to develop protocols for stroke participants. More recently, Jules Gomel, who is a PhD student I'm co-supervising with Fred Dehaye from Isae Super we're interested perhaps whether investigating to what extent textured flicker composed of small Gabor patches can be suitable for SSVP. So I'm not working only on motor imagery. Sometimes I'm diverging towards SSVP. And in particular, Jules investigated the neurophysiological signatures to identify the most suited flicker frequency, and he demonstrated that the classification accuracy was highest when each stimulus and similar types was used in its own optimal band. For instance, here for the Gabor, we were better within the theta band, whereas with the plain stimulus, we were better between 14 and 18 Hertz. And so this kind of work could pave the way to a more user-centered multi-target SSVP BCIs. And on my side, given my previous life doing my PhD, I'm still in touch with my former PhD supervisors who founded the Map for Health company. And I'm interested to see whether this new sensors, MED sensors actually, could be relevant for the BCI and to try to bring the BCI outside the laboratory. This for the hardware part. Regarding another topic that is dear to me, that is actually how to guide the feature selection during the experiment, I would like to briefly mention several aspects. First, the work led by Cassandra Dumas. She is a PhD student I'm co-supervising with Nathalie Georges at the ICM, and she would like to optimize our choice of the common spatial components, because usually we're trying to our main judgment criteria relied on the best contrast between the task. But sometimes, as you can see here on this picture, you can have some ocular artifact that remains after selecting these components. So Cassandra is working in developing an automatic method that also take into consideration the neurophysiological pattern to make sure that the component that's going to be selected has the best neurophysiological signatures linked to the task performed by the participant. And all these developments we're making to identify new features or to make sure that the feature we're selecting makes sense is meant to be integrated within our software called HappyFit. The main developer is Arthur Desbois. And the aim of HappyFit is to guide the experimenter. No need to be an aficionado of machine learning or deep learning. It's actually meant also for clinician, and to guide the experimenter in extracting, selecting the features through visualization, also statistical analysis, and also to enable the experimenter to train the classifier. And in the end, the point would be to send all these parameters to their favorite BCI platform to perform the experiment. So we don't aim to replace OpenVime Typeflex, but it's more to help the experimenter in extracting and selecting the most suited features. And actually, this kind of elements brings me to a little perhaps claim I would like to make about open science. And just I would like to briefly mention a paper that's actually, in my opinion, quite relevant about open science, and that has been proposed by the core developers of MEN softwares to perform signal processing in AMIG and EEG, meaning Brainstorm, EEG Lab, Python, and Fieldtrip. And they raised some warnings about the lack of stable long-term funding for to develop scientific softwares. And also they advise for a better recognition of the main contributors that are usually, not only, but usually PhD students and postdocs. But I think that such discussion could also include data collection and sharing, because, for instance, I briefly mentioned MoeBB, that gives you access to many BCI data sets, but behind it, it corresponds a huge amount of work and recordings made by people. And I think it's also important to recognize the work done by these persons. So even as a French people, I love complaining, but the point of here is more so to find perhaps solutions, perhaps mutualization of the resources through collaboration, for instance, to make this viable. So just a small parenthesis in this presentation. So yeah, perhaps in the end, I think it's important also to thank all these collaborations, because it's true that I mostly focus my presentation on BCI application, but I worked actually on diverse number of projects beyond the BCI, because there is another life beyond BCI. And I think it's also super important to have the chance to work in an environment that is particularly stimulating and rewarding. So I would like to thank all these collaborators in France and elsewhere. And of course, I would like to thank my colleagues from the NERV group in Paris, and particularly I'm grateful to my closest collaborators here at the bottom of the slides, from whom I'm learning something new every day. So I think it's also important to reward them and to acknowledge their huge work. And with that, I would like to thank you for your attention.
All right. Thank you very much for this very interesting journey into your research. By the way, you are very much right because we are very skeptical people because I noted down for you to tell us which part is online and which is offline. Sorry for that. So maybe you can elaborate
I know you
later.
now, so this is why I
Yeah.
anticipated your question.
All right. So now I would like to ask the audience to just raise your hand if you want to ask a question. This is the React icon in the bottom at the menu at the bottom of the Zoom screen. And in the meantime, maybe I can hit you with the first question. So this neuronal avalanche concept is very interesting, and I think now I understand it better. So effectively, you see how much the previous, how much another area predicts what happens in another area a little bit later, right? Pretty much
Yeah, it's more
something like
probability
that.
of an area of activated atom
Yeah,
time plus delta, for instance, knowing that region I
exactly.
was activated before.
So the question is, this sounds to me very similar to Granger causality, right? And it is practical, so I'm not super expert on that, but it is essentially the difference between functional connectivity metrics like the coherence you said you were also using before, and effective connectivity, right? Like the DTF method. So pretty much the effective connectivity methods use Granger causality, which is based on this concept. Now, I don't know the math exactly, but the concept is you see how much a part of each segment or point in the past predicts the future. So I was wondering, uh, have you ever compared, or in any case, do you have any insight on what the neural avalanche brings more compared to DTF or any other effective connectivity method?
So just a very small thing is it's still a functional connectivity. It's more about directed functional connectivity because since you might have a different probability from X to Y and Y to X, that is why you have a difference with respect to other estimators. So it is a very good question. Here so far, to make this analysis a bit simpler, we symmetrized the matrix. That is why we don't have this investigation about the direction of the connection. But actually, we aim with Camilla to work now on symmetrized matrix, and from this point, we could properly actually compare our findings with Granger causality. So it will be like one of our main steps in the next future to be able to compare, I mean, uh, quite widely both of these approaches. We mostly compared our approach so far with PLV and imaging coherence, either for diagnosis or for BCI classification.
Okay, thank you. Is there some question from the audience? If you prefer, you can also write it down in the chat and I can transfer it to the speaker. In the meantime, maybe I can go on with my own questions. Can you go back to the slide where you were showing the improvement of the beta band?
You mean like from the power spectrum point of view, or?
Yes, here, this one. So here I was wondering, first of all, is BCI score like accuracy or
Yeah,
something
it is
something like
accuracy. That is
accurate?
why
Okay,
I say that it's not flawless. I mean, it's a way
okay,
to
no,
measure
no,
the
no worries.
performance, but
So I'm interested in the beta band. Do you remember if the mu band didn't show any improvement that correlates with accuracy in this case?
we observe it. But yeah, by the way, here is like the beta one because yeah, at the beginning I worked in the classical mu beta band, but then I wanted to see whether we could refine a bit more this definition. So I divided into two subbands, I mean, low alpha, high alpha, low beta, and high beta. Since I was not convinced about the definition from the literature,
Mm.
I used the individual alpha frequencies, the EF. So I based my definition of the frequency band on the individual alpha peak. So it enabled me also
Okay.
to adapt this definition to each participant. Just a small comment on that. So that is why here we performed analysis in alpha 2 and beta 1, but it's true that the highest time effect has been observed within the beta 1.
Okay. And here your subjects were able-bodied or people with disabilities?
Healthy participants here.
Healthy. Okay. Yeah, the reason I'm asking is because I have this anecdotal observation that might be wrong. I mean, our data sets are not infinite, but it looks to me that people with disabilities tend to learn mainly through beta, for instance, our cybathlon subjects in the past, but not only them. And able-bodied people, especially those that, go on to become exceptionally good, apt users, they tend to use and learn move features. So I was wondering if you have maybe, since you have been doing this kind of analysis, any similar observation that would be interesting to publish, if different
Uh,
populations respond mainly with different bands.
it's a very good question. In this paper, like in your image, we also provided the features chosen during the experiment. Um, it strongly depends on the participant because I remember, like from the time frequency maps we could obtain from each participant, from some of them, you have a clear synchronization within the mu band, sometimes in beta, sometimes in both beta and mu. So it was quite diverse, to be honest. But it's true that the highest group level effect I observed with these participants was in high alpha, high mu, or low beta.
All right, I see. Um, I don't see any raised hand. Okay, so
Yeah,
I will
I have a
go on.
question. Sorry. Yeah.
Please go ahead,
Thank
please.
you so much, Marie-Constance. So my question actually is related to the chain effect. In your case, you use like two categories, like hand and the rest. So you observed the improvement. So my question is that, for example, when you consider more categories, like three or four categories, is it possible the improvement is larger? Because as you know that if we perform like four or five categories, it's quite difficult to the participants. So it means there's baseline. I mean, before the training is quite low, the performance is quite low. So after very short like session training, their performance can be improved more compared to the two category because two category usually is easier. So my question is that do you consider like to explore like the number of the categories effect on the performance improvement? So my assumption is like that if we challenge our participants, maybe the improvements might like improve more for the more challenging tasks.
It's a very good question. I guess that regarding the higher number of tasks, I mean, to be also a question for Simit, given his experience from Cyberton for each participant, sometimes when they have to pilot like an avatar, they have different directions to take and it corresponds to different labels. But in our case, they were BCI naive, and it's true that we have only two labels. You are perfectly right, but performing a motor imagery is not so easy actually, because behind the idea of motor imagery, I mean, many things can happen, and in particular, it depends a lot on the instruction you're going to provide to the participants. In our case, we made several choices, again, to ensure two things. First, that we could observe a tuning effect, and secondly, to make the participant a bit more active in the training process. For that, at the beginning, during the first session, we just asked them to perform motor imagery of the right hand, nothing more. Again, behind this, you could imagine many movements and on many types. For instance, is it visual? Is it kinesthetic? Is it grasping? Is it in terms of timing? Is it like a very slow movement, or is it like several graspings? It was open for them. But the idea was for them to be like the actor and to find their own strategy at the first session. And then depending on the performance, if they performed well, we leave them alone and they were up to pursue with their own strategy. If they found some difficulties in performing that, we help them, for instance, in providing more information regarding the type of motor imagery. Either by providing some more details, such as performing a kinesthetics or imagining feeling during the movement. Sometimes, I mean, usually it's easier and it's better to do this kind of motor imagery. And also, after the third session, if we observe that we are still struggling at this time, what we also did was to ask the participants about his or her hobbies and to find a strategy that is linked to their own memory. For instance, I remember that one of the participants had quite strong difficulties in performing the motor imagery. So we know that he loved hiking. So, okay, let's imagine an action and a strategy linked to this hobby, and it worked. So even behind the idea of only 2 tasks, you can make their life a bit worse.
Yeah, so it's quite challenging for some people to imagine the actions. So as in your experiment, you ask participants to imagine the right hand. Is it because most of the participants are like right-handed?
They were all right-handed. It was one of the inclusion criteria to avoid like variabilities between participants.
Yeah.
Even though, depending on the participant, even though they were right-handed, they may be less lateralized, meaning that the signature perhaps it be more closer to the center, like CZ or CPZ, instead of having a clear pattern within the left hemisphere.
Yeah, thank you so much.
Thank you.
May I jump
Yeah, please go.
With a quick question? Thank you very much, Serafim. I just have like a doubt in terms of because I missed a little bit of the beginning of the talk. With this Riemann geometry representation and geometry system, I see it in 2 ways because I work with pilots before. So I don't know if it's in the way you collect the data. So it's like having a sphere in the head. No, I mean, where you can have like the representation and the collection of the data under this manifold expression, or if it's more like when you have like a square, no, like a napkin.
Okay, napkin. Okay,
yeah, it's a napkin.
There's no drawing.
That's your Cartesian map, and you roll up like a straw, and you just like connect the endpoints, and you have like a toroid, and this data has a second level or hidden level of a representational system. So it's because I work with pilots and I have a little doubt, and congratulations. I think it's fantastic. It's one of the few words I've seen correlating these two points exactly.
So yeah, so let me just go back there. So it will perhaps more linked to your first hypothesis. And then once you have this curved manifold, you can also project them into a tangent space. And usually you can also perform this classification within this tangent space. The idea is also to reduce dimensionality of your problem, because at the beginning you have a full covariance matrix, and then you're going to project them as a single point. So one point would refer to a single covariance matrix. Right. So you have 1 point per trial, and then by comparing the distances towards the barycenter of each task, you can infer, for instance, when you have a new trial, whether you're closer to barycenter of task 1 or task 2.
Excellent. Thank you.
You're welcome.
If there is any other question, please just unmute and ask. In the meantime, I have a couple on the practicalities. So first, I was wondering are there any trade-offs? Do you discount something else in order to be able to bring these connectivity features? And especially when you do with MEG, which means you will have much more pairs, right? To work on. So is this nowadays possible to do fully online, or for instance, you have to discount the feedback refresh rate? So, so that you give time for the computation.
There are different approaches actually, because as you said, you can have a full matrix, but then you will have the question about whether all the elements are meaningful for the classification. Because it's a bit like when you're estimating the power spectra from all the electrodes from your EEG cap, are they all relevant? Perhaps a small subset is sufficient. So there are two approaches that can be adopted. First, you can apply some dimensional reduction analysis, and it's also what we have done also for the Vaughan approach. I try perhaps, okay, if I have some time, I can show you a bit, but what I also tried is to develop an algorithm that tends to first perform some statistical tests, let's say, okay, task 1, task 2. And based on the p-values of this t-test, I can count how many times a specific node was involved in a significant, in age in which you have a significant effect between the two conditions. And in this case, I could create a kind of ranking of the region of interest. And based on that, thanks to that, I could identify a subset, like a submatrix, in which I could also apply the Vaughan approach. So it is the first way to reduce the base dimensionality of your problem. And a second way would be also, instead of working with the full matrix, you could extract the topological properties. For instance, with node strength, instead of having the full matrix, we'd have one single vector. So could be of interest. And we could use that perhaps a bit more sophisticated
networks. I meant even before that. So then I understand that the computation itself of the connectivity is not particularly computationally expensive. Okay, good.
No, thanks to Arthur in our team. He worked hard on that actually to make this possible.
Okay, so you optimize the code probably. All right.
Yeah.
And but I mean, it's doable, because I remember we had a colleague back in EPFL when I was there that was working on that, and I remember it was challenging, but of course that was 15 years ago.
But also perhaps just a small comment, because there is the computation thing. I mean, whether you can actually compute in a very fast way these matrices, but then there is also the question of the robustness of your observation. Because whether when you have like a buffer, whether between buffer A and B, you have some similarities within your observation, or whether you have like some completely different matrix, and in this case, your feature is not so relevant. So that is also this question behind the robustness of your observation.
And maybe a quick final one. So you showed that when you combine EEG and MEG, you have an improvement, but MEG alone doesn't seem to provide a competitive edge at this moment, and especially OPM. Was that OPM MEG or was it a standard one?
No, it's the current MEG one. Perhaps my answer would be it depends, because for instance, and perhaps it'd be like the behind the scene thought, this kind of paradigm I chose here was based on one observation, because a dear friend of mine did not perform well during the experiment. He had like 40% of performance. We were a bit sad of that, but it was life. But then when I compared his brain signatures in MEG and EEG, I couldn't observe any desynchronization, whereas in MEG, it was crystal clear. Meaning that, to answer your question, I would say it depends a lot on the participant. That is why I tried as much as possible to adapt this pipeline to each participant. That is why we have some weights, because it can be different from one person to another.
Okay, that's what I'm asking, because I would expect MEG to be at least a little better thanks to the higher resolution, even
with a gradient. Yes, with the gradiometers, you're improving actually a bit with respect to the EEG, because we're removing some noise with respect to the EEG.
Okay, so if there is no other question, I think we can wrap it up here. Thank you very much, Marie-Constance, and we are
Thank
looking
you.
forward to, reading more of your research in the years to come. Bye-bye.
Thank you very much. Goodbye.
Thank you. Thank you.